Let us bridge the conversation here from last week where we talked about the cellular mechanisms of aging and talk about the mitochondria in specific detail as far as how mitochondrial function is thought to change with aging or influence aging. So by the end of this presentation, we should have an understanding of the mitochondrial changes that alter cell function over time, as well as talk more about oxidative stress and oxidative damage and where that comes into play as far as mitochondria and aging. And as we talk about the mitochondria, it's really important to speak about free radicals and oxidative stress that they accumulate in cells. And we can kind of start by looking at this image here, which shows us how all of the body systems can be influenced by this free radical stress, this oxidative stress that's being put on our cells, specifically at the level of the mitochondria. And so we can see the functional and the physical symptoms and diseases and pathology that arises, but we're really going to delve into what's happening at a cellular level that then gives rise to some of these functional systemic consequences. So we're continuing here to look at cellular organelles, specifically the mitochondria. We'll talk about the mitochondrial theory today and also incorporate the idea of oxidative stress. And I'm also pointing out here the kind of multifactorial process that's involved in cellular aging. So we had this image last time, which kind of gave us a sense of the various players involved. So we talked about telomeres at length and the cellular clock last week. We also spoke a little bit about signal transduction as it applied to hormones and sort of the blunting of the sensitivity of some of our signal transduction pathways with aging. And then we spoke a little bit about the cell cycle regulation and the cell's inability to regulate its own replication and repair process. We've talked a lot about DNA as well. So the mitochondria kind of just fits into that overall discussion of the cellular components to aging. And it really ties nicely with these cellular aging theories. Although it kind of differs as far as oxidative stress, it really fits with that multifactorial description of aging. Now, the oxidative damage and the mitochondrial aging theory are essentially the same theory. They were established by the same scientists, Dr. Denham Harmon, throughout various periods between the 1950s and 1980s. He then refined that theory to become the mitochondrial free radical theory, but it's essentially two perspectives of the same idea. So the first thought was that there's oxidative damage accumulating in our cells that could damage DNA, that could damage proteins, that could damage fats and other biomolecules. And then the other side of that theory, as he refined the theory later on, was that this was specific to the mitochondria. So mitochondrial DNA was being damaged, which was resulting in aging and some of the aging, symptoms and disease processes that we see, as well as altered mitochondrial function. So not only are we damaging the mitochondrial DNA, a lot of the DNA in our cells are made in the mitochondria and we're depleting that DNA, but we're also accumulating this oxidative stress that depletes the mitochondria's ability to function effectively. And we know how crucial mitochondria are to the energy needs and the energy production of our cells and our entire bodies, truthfully. And so let's talk a little bit more here about that oxidative damage or oxidative stress. It stems from the mitochondria. Why? The mitochondria is what we call the powerhouse of the cell. I'm sure you would have heard that term before. This is where most of the energy production of cells, as far as ATP production is made, right? The electron transport chain via oxidative phosphorylation that happens across the inner mitochondrial membrane. This is where most of the oxygen is utilized. Remember, oxygen acts as the final electron acceptor, in order to keep that electron transport chain going. So oxygen is used most in the mitochondria, energy is produced most in the mitochondria. And so this is where the mitochondria comes into the conversation as far as oxidative damage. Now, oxygen is acquired. We know it's vital to life, but it is damaging, right? It's a free radical at the core of it at high concentrations, and it can be toxic to plants and animals as well. Pure oxygen can be damaging to the lungs, especially for prolonged exposure, and it could even damage our alveoli, which are the small functional units of the lung. Why is it toxic? We'll talk a bit about the structure of oxygen, but it basically grabs or hogs or creates this unstable environment around our other atoms and cells. And so it can damage by causing that free radical interaction between some of the free electrons that are on the oxygen molecule itself. So what are free radicals? When we say that oxygen is a free radical, and this nature of oxygen is what can make it quite toxic, a free radical is a chemical or a molecule that has an odd number of electrons. So there isn't this equal sharing of electrons around the molecule. And so the unpaired free electrons are completely reactive. They're highly reactive. They're trying to combine with other molecules in order to grab or share some of those electrons. So this creates for a very unstable environment with these types of molecules, where they can be quite reactive and they can be trying to break some of the covalent bonds of some of the molecules that are in contact with them. And so what this leads to is essentially cellular damage, right? We create this unstable environment where oxygen is trying to grab electrons from other molecules that are in the cell. And so that creates this unstable, unhealthy interaction between oxygen and some of the other chemicals and molecules in our cells. And we can see here what a healthy atom looks like. It's got equal sharing around the molecule, whereas a free radical, such as oxygen, has unequal sharing. And so we've got these free components that are trying to make interactions, or basically scavenging the cell for other electrons that they can grab off of other molecules. And so this generates this unstable, cellular damaging, toxic event in our cells. And this is where antioxidants come in. They combat this free radical scavenging nature of oxygen, and they offer additional electrons with which those free radicals can pair. So they create a bit more of a stable environment and prevent some of those free radical interactions between oxygen and some of our other molecules in the cell. And that's the benefit and the purpose of antioxidants and how they've become so popular, especially as it pertains to anti-aging and combating the oxidative stress that's put on our cells by oxygen. So in this image on the right, we can see some of the sites of oxidative damage. So the DNA is a really important one, specifically mitochondrial DNA. So having those free radicals, once again, coming into contact with our DNA, this can alter important properties of our DNA, which can alter gene expression altogether. This can also deplete the amount of ATP that's produced as damage to that mitochondria and the mitochondrial DNA occurs. As far as proteins, we can have oxidation of proteins, which essentially interrupts their functional ability. And we know proteins have a whole host of functions throughout our bodies. They are hormones, they are receptors, they're enzymes. And so oxidation of hormones can lead to defective functioning of our hormones, as well as deactivation of hormones, and sometimes activation inappropriately of hormones as well, of proteins as well. And then finally, lipids. So lipid peroxidation is the process that occurs when there's damage to our lipids. We know that lipids are an essential part of the plasma membrane. Remember, the phospholipids that make up the phospholipid bilayer are lipids themselves. And so when we have this peroxidation, which again is that free radical interaction between oxygen and our lipids in the membrane, we can see damage to the membrane, which again overall results in cellular injury. So these are some of the various ways, biomolecules, organelles that are involved in oxidative stress. And these cellular organelle changes of oxidative damage are what result in the functional changes that we can see on the left. So we looked at this image last week, and we bring it up here again because these are the overall physical consequences of some of the oxidative stress that's happening on a cellular level. So we can see damage to the eyes, damage to the heart, skin, basically every single organ system can show signs of oxidative stress, of accumulation of that oxidative damage by some of the cellular processes and cellular damage that we see here on the right. And so over time, this scientist, Harman, he revised his theory in 1972. His initial theory was that oxidative stress, oxidative damage occurs equally throughout the cell. And so we can see damage to our cellular DNA, we can see damage to our other cellular organelles, as well as to our cell membrane. In 1972, he revised this theory to suggest that specifically the mitochondria is an active part and is most susceptible to some of the reactive oxygen species and most susceptible to that oxidative damage and oxidative stress. His revised theory suggests that the mitochondrial DNA is not as protective, is not as resilient against some of this oxidative stress as the rest of the DNA in the cell, as the nuclear DNA is. And he also suggests that some of the non-mitochondrial components in the cell, so maybe the plasma membrane itself of that cell, maybe some of the other organelles in the cell, have a protective measure, but the organelles, the membrane of the mitochondria itself, the DNA in the mitochondria itself, does not have as well developed protective mechanisms from that oxidative stress. And so he suggests here that mitochondrial DNA damage, mitochondrial membrane damage happens at a higher level, at a faster rate than nuclear DNA damage and nuclear membrane damage does. So basically the mitochondria is more susceptible, is weaker at protecting itself against the oxidative stress. And so we really see a focused accumulation of damaged DNA and damaged cellular components and damaged functioning at the level of the mitochondria than we do anywhere else in the cell. And this is what really makes the mitochondria important as we talk about oxidative stress and as we talk about aging. His theory, his revised or his evolved theory goes on to indicate that because the mitochondria is where most of the oxygen is used, because it's also the area where we've got this passing of electrons through the electron transport chain, we also see that there's more of a likelihood for these electrons that are being passed and utilized at a higher level in the mitochondria to escape, to come in contact with water, and to form more of these reactive oxygen species, which can have even more of a harmful, damaging consequence within the mitochondria itself. So not only is the mitochondria more susceptible and less efficient at protecting itself, but it's also more exposed because of all of what's happening in the mitochondria as it pertains to the cell's energetic requirements, ATP production, the electron transport chain, and the use of oxygen. And so he also goes on to say in this revised theory that these damaged cells, these damaged cellular components rather, are more likely to produce more ROS byproducts or more reactive oxygen species byproducts that will just kind of potentiate or augment this oxidative damage or oxidative stress. So basically, damaged cellular components create more damaged cellular components in this perpetual cycle, which leads to overall deterioration of the mitochondria, which eventually spreads to overall deterioration of the cells. And then that becomes even more broad or more global in terms of the tissue level, the entire organ system. And that's what results in some of those big functional, those big systemic consequences that we see in terms of various systems being affected. Now, some of the actual consequences of mitochondrial DNA damage are impaired ability to express genes because of damaged DNA, inability or a decrease in the ability of mitochondria to be able to replicate themselves at a rate that can keep up with the energetic demands of cells, especially things like muscle cells and liver cells that demand a lot of ATP and a lot of mitochondrial function. There's also a decreased amount of intact mitochondria, so we pretty much deplete our healthy mitochondria through this type of oxidative damage. And then that generally results in overall organ system dysfunction. So essentially, if you're depleting your healthy mitochondria, if mitochondria that are functioning are also not able to replicate fast enough, and if mitochondria that are functioning are also not able to make new DNA and express new genes, then we see an overall decline in the entire function of those organs and that organ system. So now that we've kind of talked a little bit more about the mitochondria and we kind of added this or compounded this to our discussion last week as far as telomeres and other organelles in the cell, I want you to reflect on some of the questions here this week and share your thoughts, as usual, supported by literature, supported by examples and experiences of your own personal examples, personal stories, in your discussion post this week.